Hi everyone, today we're going to finish up talking about acids and bases. So like we said last time, when you're doing an acid or a base reaction, you want to know how much acid or base is there, and you can take the advantage of the fact that when you mix an acid with a base together, you'll get a chemical reaction, and that during that chemical reaction, the acid and the base will react with each other and neutralize each other to create water and usually a salt. So if you had a sample and you didn't know how much acid was in it, you could add base to it until all the acid gets used up, and then you could figure out how much acid there was in the sample to begin with. Or if you had a sample full of base and you didn't know how much base there was, you could add acid drop by drop until all the base is used up and the solution becomes neutral, and you could figure out how much base was present in your sample. So as long as you either have a standardized acid or a standardized base where you know how many moles per liter or moles per milliliter are present, you can do this process of a titration to figure out how much of an unknown acid or base is present. So this is another example of doing one of those types of calculations where we're being told that in this particular case, we have a sample that has H2SO4, which is an acid. So we have an acid sample here, but we don't know what its concentration is. It has an unknown concentration in terms of molarity, but we do know that we have half a liter of this sample, 0.5 liters of this sample. And when we took 0.5 liters of that sample and we added base to it, and in this case we added NaOH, and the NaOH we added was 0.1 moles per liter concentration, and it took 0.75 liters of this 0.1 mole per liter concentration NaOH to react with all of the acid that was present. And if we look at the reaction that was going on here, we can see that we have a reaction where there's 1 mole of H2SO4 reacting with 2 moles of NaOH. So there's going to be a 2-to-1 ratio when we try to figure out what the concentration is for these samples. We know that this is a reaction where they're both going to occur. This is a titration, so NaOH acid reacts with, and base reacts with H2SO4 acid, and that's going to go until we run out of NaOH, and we know we needed to add 0.75 liters of the NaOH until we ran out. So we can figure out how many moles of NaOH are in 0.75 liters. We'll just multiply the volume, the 0.75 liters, times the concentration, the 0.1 moles per liter. And in that case, my liters here of NaOH cancels with my moles per liter of NaOH. And it turns out I had to add 0.075 moles of NaOH to get my acid to react with all of the base. So to get all of that acid to react, I needed to add 0.075 moles of base of 0.08 of NaOH. But we also know that for every Na acid that's present, you actually need 2 NaOHs. So one H2SO4 reacts with 2 NaOHs. So if we want to figure out how many moles of H2SO4 are there, we'll use the moles of NaOH, the 0.075. So we take our 0.075 moles of NaOH, and we know that for every mole of NaOH, we actually have 2 moles of NaOH reacting with 1 mole of H2SO4. So we write that down here to show that there's 2 NaOHs reacting with 1 H2SO4, and that way our moles of NaOH cancel with our moles of NaOH, and we're left with a number of moles of H2SO4. So we have a certain amount of H2SO4 present. Turns out that this particular sample had 0.0375 moles of H2SO4 present in that half liter of volume. So now we have our volume, our sample, we have our concentration of NaOH in terms of moles, we also have our volume, so we can know that there were this many moles of NaOH in a certain volume, and we can figure out what the concentration was. So 0.0375 moles of NaOH divided by of H2SO4 divided by the volume of H2SO4, the 0.5 liters, which means that our concentration for this particular sample was 0.075 molar H2SO4. There were 0.0375 moles of H2SO4 in half of a liter, and 0.0375 divided by half comes out to 0.075 moles per liter. So that's a way you could figure out the concentration of an acid by using the concentration of a base that you've added to it through a titration type reaction to figure out how many moles of acid are present after you've added the base. And this is just one kind of final example, kind of showing a multiple choice version of this same question. So here we've got a few different things added. We have half a milliliter of acetic acid, so that's the volume of the acid. So we know that there's a certain amount of acid that was present. So we can write that down first, that we have half a mil of acid, and also that we added NaOH, and we added 21.6 milliliters of the NaOH. So we have 21.6 milliliters of the base, 50 milliliters of the acid, and we also want to know what the concentration of the acid is. We don't know what the concentration of the acid is. They haven't told us, but we can calculate it because we do have the concentration of the base, 0.512. So we have the concentration of the base, we have the volume of the base, and we have the volume of the acid. So using those three things, we can figure out how many moles of base were present, how many moles of acid were present, and then we'll divide the number of moles of acid by the volume of the acid, and that'll give us the concentration of that acid. So the first thing we got to do is figure out what the concentration of the base was, how many moles there were of the base. We know that we had 0.512 moles of a sample per liter, and we have 21.68 milliliters of that sample. So if we multiply the volume times the concentration, or the 21.68 times the 0.512, we'll end up with how many moles were there. So now we have a certain number of moles, that's the moles of base, and this is a 1-to-1 ratio. There's one base reacting with one acid. So if we divide this number here by the volume of the acid, that'll tell us the concentration of that acid. We know that the moles of acid equals the moles of base because this is a 1-to-1 ratio when you balance it. And then when we look at the next step, we divide by the volume. So the 11.1 number divided by 50, which is the volume, that gives us the concentration that we've been trying to solve for this acid. Turns out our concentration is 0.222 moles per liter. And if we look at that on these choices, that's choice B right here. So using this equation here, looking at the number of moles per liter, multiplying that by the volume, we're able to figure out moles. And then we can convert moles of base into moles of acid and divide by the volume to figure out how many moles per liter there were. So those are some examples of titrations that you can use to figure out the concentration of an acid solution or the concentration of a base solution by adding a certain volume of a base or an acid at a certain concentration and figuring out how many moles you added. So we've been talking a little bit about acids and bases and ways of figuring out what an acid is or figuring out what a base is, figuring out the concentration of an acid or a base using things like pH. Or if you don't know the concentration, how you can use something like a titration to figure out what the concentration of an acid or a base is. And one of the sources, if you look around at, when we looked at different acids and bases that are present in nature, we noticed that there were quite a few foods that were acidic, but also that the acid in our stomach, there's acid present there as well, which often people wonder, why is there acid in our stomach? Acids seem to be pretty not good things to have around. They destroy metal, they degrade things, they cause acid rains, and they kill fish. So why is there acid in our stomach? And also, why do people take antacids? Why do acid foods exist? And why do people get stomach aches from eating too much of acidic foods? Because there are quite a few foods that have acid in them, but even though there's acid in our stomach, that doesn't seem to cause a problem. But sometimes if you have too many acidic things, you can have a problem. So we'll talk about a little bit of all of those different things. And that comes from this idea that we need to break down foods and other molecules to get energy. So different ways of getting energy is by a catabolic process of taking high energy things like carbohydrates, fats, and proteins, and breaking them down into their building blocks, breaking them down into the things they're made of. And then that gives off a lot of energy. And then we can take all those different building blocks and we can actually build up those molecules into large materials that we might need later. So we eat a bunch of beans, or we eat a bunch of celery, or a bunch of broccoli, and it's full of plant proteins, and we want to take those proteins and we want to make our own proteins because we can't use plant proteins, but we can use the amino acids and the sugars and the fatty acids that those plant proteins are made of, break them down into the individual building blocks, and then build them back up into the proteins that we need. And in the process, we're going to get a lot of energy from that. But we need to break these things down, and one way to break these things down is to use acid. Acid is really good at actually breaking down proteins into the smaller building blocks, the peptides or the individual amino acids that we can use to build larger proteins for ourselves. So this is a really important step in digestion, is to use either strong acid or strong base. So we happen to use acid in ours, but that's why cleaning materials are either acid or base. So if you look around the house, the different things for cleaning materials are generally either really strong acid or really strong base. We use strong acid in our stomach to help break down proteins. And our stomach actually is a really interesting pump system for making acid. So our stomach has a pH of about 0.8 or somewhere around 1, which means that 0.1 moles per liter of HCl in our stomach. And the way that works is that your stomach has a series of pumps in them that basically transmit H's in. So H's go into the pump from the outside of your cells into the stomach. So this is the inside of the stomach over here, and this is the outside of your stomach. So outside your stomach is a regular cellular environment, pH 7, neutral pH, just like the rest of your body, pH 7. But the inside of your stomach is a pH of 1. So you're going from a pH of 7 over here to a pH of 1 over here. And all that's separating that pH 7 and pH 1 environment is usually just one layer of cells or one membrane of cells. So a single membrane or a single layer of cells is separating a really, really acidic environment inside your stomach from a really, really neutral environment outside. And the way that this works is that it basically pulls in protons from outside of the cell to the inside of the cell. But when it does that, that puts too many positive things inside the stomach. So the stomach has too much of a positive charge, and it doesn't like that. So what these cells also do is they take K pluses, which are also positive 1, and it kicks those out. So as the Ks get kicked out of the stomach, the H's go in, and you have a net 0 change in charge. Even though you're pulling H's in, you're pulling a K out. So the overall H difference, the level of positiveness, doesn't change. But then what happens is after the K leaves, it hooks up with a Cl minus ion, and now K plus and Cl minus ion, they cancel each other out, there's 0 charge, and then they come together back inside of the cell. So now you have a K coming back into the cell that just left, but it brought a friend with it, it brought the Cl with it. So the K leaves and it goes around and comes back into the stomach, and it brings a Cl with it. So overall what happens is you move an H into your stomach, and you move a Cl into your stomach, and HCl and that makes HCl, which is an acid. So the K is constantly kind of coming and going back and forth, left and right, but the H's and the Cl's come in. So you get this kind of overall left and right, while the H's and the Cl's are coming in. And that creates an acid environment in your stomach that's really useful for you because it lets you digest food. It's really important to have, but it's also the most concentrated gradient of all cells. So one cell is blocking a 10 to the 6 or 1,000,000 times concentration difference. The amount of acid inside your stomach compared to outside your stomach is about 1,000,000 times more intense. So there's about 1,000,000 times more acid molecules in your stomach than there are outside your stomach. And that's a very, very big difference. So 1,000,000 times more concentration of acid inside your stomach than outside your stomach. With just a single layer of cells blocking that difference. So that's why we want to keep all the acid in your stomach. We want to make sure it doesn't go anywhere. But if you have too much acid in your stomach, that can cause a problem too. And that's where some people, they have a problem if they have too much acid in their stomach. Either they have digestive problems because they don't have enough acid. That's if you have like problems digesting food, you don't have enough acid and you can't break the food down. But if you have acid reflux or any kind of high acid content, you may have problems, either ulcers or throat problems, from having too much acid. So that can either be because you as a person just generate too much, you're really good at making acid in your stomach, and it's just a very, very high amount of acid, a little bit more than you need, or if you eat a lot of acid foods. So if you eat a lot of tomatoes, a lot of juices, a lot of things with a high acid content, then your stomach may have too much acid in it as well. And if they do that, sometimes you burp or you have problems, you feel the burning in your throat, and you have heartburn, sometimes what's called. This is from having too much acid, and there's a few different ways to treat it, but one is to take an antacid. So antacids are like Tums or any of those kind of molecule drugs that you take. What they do is they react with the acid. Antacids are just bases. So they're either magnesium hydroxide or calcium hydroxide tablets. So it's basically chalk and magnesium. And if you eat a little bit of that, then what happens is the OH of the antacid reacts with the H, and you produce HOH or water, and you basically react with some of those acids, and you lower the amount of acid that's present in your stomach by doing a titration or a reaction of an acid and a base. And that causes the sample to lose some of the acid present. So there's a little bit less acid in your stomach, and that way, without all the acid in your stomach there, you don't get quite as much acid around it. It doesn't burn your throat or burn your stomach as much, or it doesn't cause as big an ulcer. But at the same time, if you have too many of these molecules, you can actually get rid of too much HCl, and you can cause either calcium poisoning from having too much calcium or magnesium, or from having so much HCl that you actually start to affect the pH of your stomach so much that you can't digest food and you have a problem. So don't take too many antacids. That's a bad thing as well. But a few antacids kind of adjust the pH of your stomach slightly so it's not causing you problems is okay. So that's kind of the idea of what an antacid is and why people may take one. So when we're talking about acids and bases, the other thing that comes up sometimes is that not every acid only has just one proton or one H for it. Some acids actually can release more than one proton or have more than one H present in them, and those are called di or triprotic or sometimes polyprotic acids. They have more than one proton. They're polyproton or polyprotic acids. So the typical acids we've been talking about so far are monoprotic. They only have one proton in them. They're things like HCl, HNO3. But some acids actually can release more than one proton. Those are called the diprotic acids, and they have 2 protons per molecule. So they'll give off 2 protons per molecule. You'll get twice as much protons from a diprotic as you would from a monoprotic. And there's even acids that are triprotic. They have 3 protons attached to them, and all 3 of those protons can come off and go into solution. So 1 mole of a triprotic acid could potentially give you 3 moles of protons, opposed to 1 mole of monoprotic only giving you 1 mole. So monoprotic are things like the HCl, the HBr, the HF, the HNO3. And you can see here, when you look at their formulas, right, there's only one H attached to that. HCl, one H, one Cl. HNO3, one H, one NO3. So those only have one proton attached to them, and that's the only thing that goes into solution there. But if you look at like the diprotics, like the H2SO4, right, there's 2 H's for this SO4 molecule. So one SO4 goes into solution, and then 2 H's go into solution. So if you had 2 moles of H2SO4, you'd actually have 4 moles of H, versus having 2 moles of HCl, you'd only have 2 moles of H. So depending on what kind of acid you have, you may have more than one proton released. And you can even have triprotic acids like the H3PO4, and again, that's going to give off 3 protons per mole of sample there. And when these acids break down, though, they do break down kind of stepwise. So first one proton comes off, and then a second proton comes off. They don't come off at both at the same time, but maybe this occurs so frequently and so fast that it looks like they both do. But generally, only one proton comes off at a time. So if you had something like the diprotic acid H2SO4, the first step is that one of the H's comes off of the H2SO4, and you're left with HSO4. So that's the first proton coming off. And then that HSO4, that second H can come off as well, leaving you just the SO4. So if we're looking at acids and bases or conjugate bases, right, the acid is H2SO4, and then the conjugate base is HSO4. And then if we look at the acid-base system in this row here, the acid is the HSO4, and the conjugate base is the SO4. That's the base without the acid attached, without the proton. But overall, what it looks like is H2SO4 breaks down to give you HSO4 and then 2 H's in solution. So either way, this SO4 is the conjugate base, and then this is the acid. So this is an example of a diprotic acid breaking down in steps. Triprotic acids can do the same thing, except they would go 3 steps. The first step is the H3 goes to H2. Second step, H2 goes to HP, and then HP goes to no H's, just PO4. So that would happen in 3 different steps, and the net effect of that step is that you get 3 protons released into solution. You'd still have a conjugate base, the PO4 minus, and you still have an acid. You just have an acid that can give off 3 different protons. So this would be an example of a triprotic acid, an acid that could potentially give you 3 moles of protons for every 1 mole of acid when you do this, but it's going to go step by step by step if it does do that reaction. Another thing that's talked about, in addition to the concentration of an acid, is acids are also classified as monoprotic, diprotic, triprotic. They're classified as what concentration, what moles per liter of H they are, but they're also classified by how strong or how weak the acid is. And strong acids have a tendency to break up completely. So strong acids, when you take a strong acid and you put it in solution, all of the H's are going to leave the acid, and you'll be left with acid as a conjugate base and nothing, and protons in solution and nothing else. So very, very strong breakdown of H acid into acid minus and H3O plus. And all the acids we've been talking about so far are strong acids. They break down completely, they give away all their protons, and they form 100% the conjugate base and the proton together for those systems. But there's also what are called weak acids. Weak acids don't do that process as well. And if you were looking at a weak acid in solution, if you took a weak acid and dissolved it in solution, you'd start with some H acid, and when you were finished, you would have acid minus like you did with the strong acid. You'd have H3O like you did with the strong acid, but you'd also have a lot of this H acid left behind unreacted. Weak acids react very slowly, they don't give their protons up as frequently, and therefore when you look later on, you still have a lot of the acid with the protons still attached to it. So if you're comparing a weak acid to a strong acid, for the same concentration, the pH will be much higher for the weak acid than it would be for the strong acid. Or if you were looking at the amount of protons in solution, there'll be a lot less protons in solution with a weak acid solution compared to a strong acid solution of the same concentration. But when we talk about an acid being as strong or weak, we're talking about its ability to break apart. We're not talking about how concentrated it is. HCl is a strong acid no matter how concentrated it is. It could be 0.01 molar HCl. It's still a strong acid even though it's a very low concentration. And H2CO3 is still a weak acid even if it's really, really concentrated, because it's not the concentration that matters for whether or not an acid is strong or weak. It's the amount of dissociation, what's called the K factor, when we'll talk about in another second there. So the ability of an acid to break down into different associate from the acid to the weak acid state, that's measured by this value K. And if you have a really, really high K value or a very large K value, that's an acid that's strong. That's an acid that goes from having the proton attached to the proton not attached. And there's nothing left over with the proton still attached in it. If you were to look at the sample after the reaction, it's all acid minus and H3O, and there's no H acid left present in a strong acid system. Versus if you look at a weak acid system, the value for K in that case is going to be very small, and most of the sample will still have the H attached to the acid. So H acid will still be the dominant species there, and there won't be as much H3O present as you would expect for a sample that concentration. And that's because those weak acids are holding onto their protons very, very tightly, and they hold onto their protons so tightly that when the proton tries to leave the acid, when the proton tries to leave the H acid, it doesn't. It sticks around and it stays attached, so it doesn't go off into solution and cause the solution to become acidic. So weak acids hold their protons very, very tightly, and that prevents the proton from leaving the acid and keeps it attached so that the concentration of H plus doesn't increase. And this is just some visual depictions of an acid-base system. So you can see here, we have a very strong acid. We have an acid with an H attached to it. And if we dissolve that in water for a strong acid system, all of the H's are going to leave the acid. So if you were to look after a few seconds, you would see all the H's on their own. And that means that this is a strong acid. It has a very low pH, and all of the acid and the base components have separated from each other. The acid is H, the conjugate base is the acid minus, and those are totally separated from each other, and that would be what we call a strong acid with a high value of K or a large value of K. If we were to look instead at a weak acid, we would see something different. We'd take our weak acid solution and we dissolve that in water. Then what we see is that a few protons come off. One acid proton pair separate. So we still have a conjugate acid. We still have the conjugate base. We still have the acid. Those have separated. But the other components of the acid are still together. They haven't separated. And therefore, the pH of this system is going to be much higher than the pH of the system over here. there's much less H plus in solution down here than there is up here because the acids are still hanging on to their H's. The conjugate base and the acid H are still attached to each other, and they're not separating the same way that they would in a strong acid system. So strong acids hold on to their protons very weakly, and that lets the proton leave, versus weak acids hang on to their proton really tightly, and that means that they keep that proton next to them and it doesn't leave and it doesn't affect the pH, it doesn't go into solution. And this would be a weak acid with a very low value of K versus a strong acid would be a very high value of K if we're looking at K values. So weak acids and weak bases, this is just another example of showing a few. And you can see here, right, HCl is what's called a strong acid. And if you were to look at a solution of HCl, you would see it's all Cl minus and H3O plus. You wouldn't see any HCl present in solution because it all breaks up because it's a strong acid. Same thing with H2SO4. You would see SO4 2 minus in solution, you'd see H3O plus, but you don't see any H2SO4 because all of that breaks apart and becomes SO4 and H3O with no H2SO4 remaining, and that's because that's a strong acid. Versus over here, down here, we can see some weak acids. Weak acid like HNO2. If you started with HNO2, you started with 2 moles of H2 and 2 atoms of HNO2. One of them stayed as HNO2 and one of them split up. And because there's quite a bit of HNO2 present, that means this is a weak acid with a low value or a small value of K, because there's not much NO2 in solution. A lot of it is still in the HNO2 form, and that's what we would call a weak acid. We can always look at these by math equations or by looking at different formulas, but the key point for this is that a large Ka value indicates that you have a strong acid, and a low or a small Ka value indicates that you have a weak acid. So there's ways of calculating the strength of an acid or a base, and it's basically the amount of H3O that's present in solution compared to the amount of acid still with the H attached to it. So it's that ratio of H acid to acid with an H that's free. And the larger that Ka is, the more likely you are to have H3O, or the more likely you are to be on this side of the equation versus the side over here with the H acid still attached. So if you have a very small Ka, then you have a lot of H acid still present, and you're a weak acid, versus if you're a large Ka, you're a strong acid. And you can see here some very large Kas and some strong acids. So if you look at the strong versus the weak acids here, sulfuric acid, nitric acid, hydrochloric acid, their Kas are very big. And then for all the weak acids, their Kas are very small, 10 to the minus 3, 10 to the minus 10, 10 to the minus 7. Those are all very, very small Ka values, and that means that things like boric acid and acetic acid and nitrous acid are all weak acids. So the large Kas are strong acids, the small Kas or the low Kas are weak acids. And this is just an example of one of those types of questions. It's asking what the strongest acid of this list is. We know that strong acids have a very large Ka, and if we look, this right here is a very, very large Ka, 10 to the 12th. So this is the largest Ka, so that would be the strongest acid. This one's the next largest Ka, C, that would be a little weaker acid. And then these bottom two are very weak acids with 10 to the minus 12 and 10 to the minus 23 Ka values. So the A and B are very weak acids, and then C is a medium acid, and then D is a very strong acid. And when we look at different acids with polyprotic acids, the polyprotic acids, different H's may be stronger or weaker. So for something like an H3PO4, H3PO4 going from H3PO4 to H2PO4, that's a strong acid. That reaction occurs very quickly, has a very high Ka, but the next two steps, the H2PO4 going to HPO4 and the HPO4 going to PO4, have very low Kas. So these two happen very weakly. So if you had a solution of H3PO4, most of it would be in this state right here, the H2PO4 state, with a little of H3O present as well. But you wouldn't see a lot of the HPO4 or the PO4 minus. So H3PO4 is sort of a weak acid because one proton comes off fast, and then the other two come off a little bit more slowly. So this acid isn't as strong as you would think it is, because it turns out the other two steps don't occur as fast. And then when we look at salts and when we look at bases, in terms of acids and base strength, one last thing that can happen is that adding salts to a solution can actually affect the pH as well. So just like a weak acid or a weak base can be affected and might not give away its proton as well, adding a little bit of salt to a solution can also affect how well an acid becomes acid or basic. And especially adding a salt to a weak base or a weak acid, that can really affect the pH of a solution. So if you have a solution with a weak acid and you add a little bit of a salt to it, you can actually make the solution a little bit more basic, because salts have both a cation, a basic component, and an anion component or an acid component. So by adding an acid to a base, you react those together and you make the sample a little bit more basic possibly. So we can use that to take advantage in biological systems where we might want to adjust the pH. So just like with our stomach, we might want to adjust the pH to not be quite as strong or not quite as low and not quite as acidic. One way to do that is by adding salts to a sample, and having a sample that has both a weak acid and a salt, you can make a sample that has a pretty strong pH adjustment. And that's what's called a buffer. A buffer is a system that resists changes in pH that basically stays at the same pH no matter how much acid or base that you add. And there is a limit to how much acid or base you can add to a buffer. That's called the buffer capacity. So after a certain point, you can't add any more acid or base to a buffer. The system just behaves like regular water. But as long as you're less than that buffer capacity, then the sample actually will resist changes in pH. So it gives you a way to prevent pH from going up or from pH from going down by having what's called a buffer solution. And a lot of biological systems are buffered so that if a little bit of acid or a little bit of base gets into them, they don't go super low pH or super high pH, because really big changes in pH are bad for a living system. But a buffer prevents some of those changes from happening. So often a buffer is a weak acid and then the salt of a weak acid, because then you have both the H acid component of the acid and the acid minus component of the acid. So you have both the acid and its conjugate base. And then if you were to add OHs, the OHs would react with the H acid, but what would it produce is it would produce more of the acid minus. And we already have acid minus in solution, so you don't change the pH of the solution at all. And the same thing with the acid minus, that can be reacted with actual acid. So you can add a conjugate base and react that with an acid. And all you do is you produce more of the H acid, but we already have H acid in solution. So having these two things together makes a sample that can prevent acid or base from reacting with it. And you keep the pH about the same no matter what. This is just showing an example of an actual buffer system. So this says HC2H3O2, and that's a weak acid. And then C2H3O2 is the acid salt or the conjugate base of that weak acid. So if we were to add OH to it, it would react with this side over here and produce more of this C2H3 sample. And if we were to add acid to it, it will react with this thing in red and produce more of the purple C2H3 sample. So this sample will withstand changes in pH from adding acid, and it'll withstand changes in pH from adding base, because both of those components are there. And if you add acid, then it'll react with the C2. And if you add base, it'll react with the HC2 sample. And this is an example kind of showing that same sample here. So this is adding what's called the universal indicator. Universal indicators are neat because they change color depending on if something's acid or basic. If something's an acid, it kind of turns a red color. If something's a base, it turns a purple color. And then if it's neutral, it stays at 7, it stays green. So by kind of looking at the sample and seeing what color it is, you can see, are you an acid, are you a base, or are you neutral? So what we did is we had this buffer sample and we added this indicator to both a buffer and to water, and they both were pH 7 to start with. But when I added acid to my samples, the water is going to react with the acid really quick, and it's going to produce lots of H3O because I'm adding H3O to the sample, and the H3O reacts with the water, and it becomes an acid because H3O acid is an acid. So that means that if we were to measure the pH of the sample or the amount of H3O that's present, we would see a lot of it because we just added it. But if I added that same acid to my buffer, what happens is the H3O would react with the C2 component, and it would all go away and become HC2. And there is no H3O now because all this H3O is reacted with my Ca2H3, so the pH doesn't change because the pH doesn't get affected because there is no H3O present in solution. It all reacted with my C2H3. And because all the H3 got used up, there's none left and the sample stays at pH 7 because this is a buffered system. So my buffered system can withstand an addition of acid. So if I had a lake, or if I lived in a lake, and I was worried about acid getting into the lake and affecting the system, if the lake was buffered, the pH of the lake wouldn't change. It would stay at 7 versus the lake going down to pH 4 when that acid got dumped in. And this works for bases as well. So with the base system, if I add base, my water is going to go from a neutral system to a base because I've added a lot of OH to the system. So by adding all this OH, it causes my sample to go from acidic or neutral to basic. But if I added that same amount of OH to my buffer system, what happens is the OH reacts with the H2, HC2 sample, and all of the H gets used up. All the OH gets used up. It reacts with all of my HC2, and what I'm left with is C2H3, but there's no base left over. There's just water. And because the base didn't got all used up, there's no free base present, my sample stays at pH 7 still. So the pH doesn't go up when I add base, the pH doesn't go down when I add acid because it has this buffer system. It has these pieces that'll react with either the acid or the base, and it keeps the pH the same. So we can figure out actually the pH of a buffer. There's a little equation that you can use if you know what acid you're using and what the strength of that acid is or what its Ka value is. You can actually figure out how strong the buffer would be and what the buffer's pH would be. And the equation for doing that is this equation right here, that the pH of a buffer is equal to the pKa, and that's the negative log of the Ka. So the negative log of the Ka factor plus the log of the concentration of the A form of the acid without a proton, and the acid with a proton. So if you have an acid there, and you have some of the acid that's in the HA form, and then you also have some of it that's in the A form without the H on it, or the conjugate base, just the ratio of the conjugate base of that acid to the acid, the log of that, and then add the pKa value, which is the negative log of the Ka. So using that, we can actually figure out what the pH of a buffer would be. So if we wanted to design a buffer, we could figure that out, or if we had a buffer, we could figure out what its pH is. So you can see here by choosing all sorts of different types of components or different types of acid, you can get all sorts of different pKa's because every acid has a different pKa. So for instance, acetic acid has a pKa of about 4.7, carbonic acid has a pKa of about 6, nitrous acid has a pKa of about 3. So depending on what you wanted your buffer to be, you could pick different pH samples. Whatever you want the pH of that buffer to be, you could pick a different acid, set up the ratio of the acid to the H acid, and you could have a buffer at any pH by adjusting which acids you have present. So this is just a practice calculation. So again, we have a buffer, and this buffer is made of two different things. It has 10 moles of HCO3 and 2 moles of NaCO3. So we have an acid. The acid that we're working with is HCO3, and that's the acid form. And then the A form, so this is HA. And then there's an A form, which is just CO3. And then that would be this one over here, the NaCO3. So if you look at the NaCO3, that was 2 moles in 1 liter. And then the HCO3 was 10 moles in 1 liter. So we can figure out the concentration of the HCO3, the HA component. We can figure out the concentration of the CO3, the A component. And then we can figure out, using the pKa, they gave us the pKa is 10 to the negative 10.25. So we can plug that all into that equation. The pH of a buffer is the pKa, 10.25, plus the concentration of the acid. Acid concentration A minus is 2/1, divided by the concentration of the H acid, in this case 10/1. So we can put all those in. So we know the acid is the CO3, and then we just have to figure out what the concentration is. So CO3 or NaCO3 is 2 moles in 1 liter. and we can figure that out to be 2 moles per liter. And doing the same thing for the H acid form, which is the HCO3, which had a concentration of 10 moles per liter, given from the problem here. We see there's 10 moles per liter of HCO3, 2 moles per liter of NaCO3, and then we have everything we need. So we have the pKa, we have the A- concentration, and we have the H acid concentration. So we plug all those in. The pKa is 10.25 plus the log of 2 divided by 10. And if we put all that into a calculator, we get 10.25 minus 0.7, and then that comes out to 9.55. So the pH of this buffer of 10 moles of HCO3 in 1 liter plus 2 moles of NaCO3 in 1 liter, so taking those two samples, mixing them together, that would give you a sample with a buffer of 9.55 pH. So if you wanted a sample that's pH was going to stay around 9.5, mixing a 2 molar solution of NaCO3 with a 10 molar solution of HCO3 would give you a solution that has a pH of about 9.5, no matter how much acid or base you add to it. So this is just another example here, doing that same calculation. We have two different concentrations. We have concentration of KNO2 and a concentration of HNO2. So nitric acid is H plus NO2. So that's the H acid form right here. That's going to be this one down here. So we know the H acid down there was 0.1, 0.1010. So that's our H acid. And we also need the concentration of the acid minus, which would just be the NO2 without the H, or in this case, the KNO2. There's no H on KNO2. So the top value for that, 212. So we have the log of 0.212 divided by the log of 0.101. And we would also need the pKa. The pKa is 4.65. So 4.65 plus the log of 0.212 divided by 0.1. And if you were to put those into the calculator, you would get a value. You would get an answer for that, and you would pick the answer from there. So buffering is really important. So being able to figure out what the concentration of a buffer is, being able to figure out what a buffer's concentration is, and figuring out how much of a sample has a buffer pH, what the pH is going to stay at, is important. And this is really important for all sorts of biological molecules, all the different reactions in your body. A lot of those are facilitated by proteins or enzymes, and they have a pH where they work, and they have pHs where they don't work. So there's a narrow pH range where these molecules work well, and then there's a larger range where they don't work at all. So you don't want to be outside of the optimal pH range. You don't want to be in any of these areas over here because the molecule won't work anymore and the reaction won't function anymore. So it's really important for biological systems that you be at the right pH and that the system stay at a buffered pH, so the pH doesn't change and the sample doesn't get degraded and your cells don't get damaged. So that's done in biological systems in your body and throughout all sorts of living creatures by having a lot of buffers. And they do that by having levels of salt and levels of conjugate acids and bases together in those cells so the pH stays the same. And one really good example of this is your blood. Your blood is buffered to be about 7.4, and that's pretty much the pH range it has to stay at. If you go much lower than 7.4, if you go down to anywhere below 6.8 or lower, which is only half a unit lower, that will be enough for is fatal. Your body won't be able to transport oxygen anymore, and you won't be able to breathe. No matter how much you breathe in or out, you won't be able to breathe in and transport the oxygen. And the same if you go above 7.4 by only half a unit and get above 7.8 or higher, the pH gets too high and the reaction stops working again. So there's an optimum pH where the system works really well, and then anything outside of that range is bad. So you don't want, if there's any extra acid added or any extra base, you could easily go below 7.4 or above 7.4. So to prevent that change in pH, blood is buffered. And by buffering the blood, the reaction is able to stay at a constant pH around 7.4. And then even if a little bit of acid gets into the blood or a little bit of base gets into the blood, the pH stays at the same. And then the reactions don't get affected by that because there's buffers. So buffers are a way of keeping a system at a constant pH, which is really good for biological living systems because that keeps them at the pH that keeps them at the right reaction for all their reactions to occur. And your blood actually does a pretty complex system. It uses carbonic acid, so it uses CO2. So when you breathe in oxygen, you also generate CO2 as a byproduct. And that byproduct actually reacts with the blood in your body. So the dissolved CO2 actually reacts with water and produces this HCO3 molecule. So you produce H2CO3 from reacting with the blood. So interacting with CO2 and water produces this H2CO3 molecule, but then that H2CO3 molecule acts as a buffer. H2CO3 can react with any base that may be present in your blood and produces HCO3 and then water. So you don't get any base getting introduced into the blood. The base reacts with the H2CO3. And then if there's H2CO3, there's also HCO3, and that HCO3 can react with the acids that may be present in your blood, and that prevents the acid from accumulating in the blood, and it keeps the balance between the H2CO3 and the HCO3 pretty good, and that makes a buffered system. So your body buffers the pH of your blood by having a combination of H2CO3 and HCO3 in the blood, so that if there's any extra molecules of base, they can react with the H2CO3 and get neutralized, or if there's any extra molecules of acid, they can react with the HCO3 and become neutralized, so there's no net result of acid or base in the blood. But because we rely on the CO2 from our breathing to produce the carbonic acid, the H2CO3 mixture that buffers our blood, the amount of air that we breathe in and the amount of air we breathe out is actually really important for maintaining the pH of the blood, because that keeps the buffer components that we need in the right ratios. And what can happen is if somebody gets nervous or upset, then they can start breathing more often and they start breathing very rapidly. And that can actually cause problems, because the way that you normally breathe versus the way they're breathing when they're upset can actually disrupt the amount of H2CO3 that's present in the blood, because there's less carbon dioxide in their blood. They're breathing out lots of carbon dioxide when they hyperventilate, and that can cause problems. And people, if they start to hyperventilate, they'll eventually pass out and faint and hit the ground. But if someone's starting to hyperventilate, you may see on TV shows, you may have seen this in various settings, that somebody will hand them a paper bag and say, breathe into this bag and you'll feel a little better. And people always ask, well, why do they make you breathe into a paper bag? If you're breathing, you can't do it with a plastic bag, that would be very bad, but you can do it with a paper bag. And why does that make them stop hyperventilating and make them feel better? And it has to do with buffers and breathing. So during normal activities, you breathe in a certain amount of air, you breathe out a certain amount of air. You breathe in a specific amount of oxygen, that oxygen gets converted into CO2, you breathe out a certain amount of CO2. But if you breathe more frequently, you can change that balance. Normally you breathe in a certain amount of oxygen and you breathe out a certain amount of CO2. And you breathe out about the same amount of CO2 with each breath. But if you're only breathing 5 times a minute, that's a certain amount of CO2. If you breathe 10 times a minute, that's twice as much CO2. And when somebody hyperventilates, they're breathing out much more frequently than they breathe in. So they start to breathe out lots and lots and lots of CO2 because they're exhaling so frequently. And as they exhale more and more and more, they start breathing out more and more CO2, and the blood level of their CO2 drops. The amount of CO2 in their blood drops. That causes the balance between the amount of H2CO3, that drops as well, because the amount of carbonic acid, the H2CO3 level, is dependent on how much CO2 is in the blood. And as they lose more and more CO2, breathe out more and more CO2, they start to drop the amount of H2CO3 that's present, and they start to affect the amount of buffer that's present in their blood. Because they lose more and more and more of this acid component, they start to reduce that amount, and it starts to actually change the pH. Because remember, we said before, we can figure out the pH of a buffer by figuring out how much of this H2CO3 component there is and how much of this HCO3 component there is. So the ratio of HCO3 to H2CO3 controls the pH of your blood. And if you start getting rid of all the H2CO3 from exhaling really, really frequently and exhaling a lot, then you start to change the pH of the blood. And you can do a quick calculation here, and you could do, 5 over 5, and then do 5 over 4, and then do 5 over 3, and you'll see as the amount of H2CO3 goes down, the pH goes up. pH goes up higher and higher and higher. And as the pH goes up, oxygen can't bind to hemoglobin very well. Hemoglobin works at pH 7.4, great for transporting oxygen. And as the pH goes higher and higher, hemoglobin in our blood can't carry oxygen very well. So we can become less able to carry oxygen, and that means there's less oxygen in our blood. So even though you're breathing in very frequently, as you breathe in and out, you start to have less oxygen present in your blood, even though they're breathing in a lot, because the blood itself can't hold the oxygen anymore. The hemoglobin loses its ability to carry oxygen because the ability to carry oxygen is pH dependent. As pH goes up, oxygen transport goes down. And you can see that on somebody. If you were to put somebody in a CAT scan or an MRI or a PT scan, you could actually look at how much oxygen's in their body, and this was looking at how much oxygen's in their brain. And you can see the red here numbers, those are good. Red is good in this case. Red, orange, white, those yellow colors, those all mean that there's a lot of oxygen present. And you can see here, this brain's got a lot of oxygen going to it during normal breathing activities. But as this person starts to hyperventilate and breathe faster and faster and faster, they start to lower the amount of CO2 in their blood. Lowering the amount of CO2 also lowers the ability of the blood to carry oxygen, and there's less oxygen in their brain. So their brain starts to go from the red side, the high oxygen side, down to the blue-purple low oxygen side. And as their brain gets less and less oxygen, they start to get lightheaded and they may start to pass out. And if you ever had somebody, when you start to get upset, that tingly feeling you start to feel, that's because you're not getting as much oxygen as you should. So if somebody gets really, really upset, they start to breathe more and more frequently, their blood CO2 drops, that increases the pH of their blood and makes it harder for them to carry oxygen to their brain and to the rest of their body. And if they keep this up for a long enough time, they can actually pass out. So why do we use a paper bag if somebody's hyperventilating? And the reason for the paper bag is that the paper bag kind of acts as a little container to hold some of the oxygen in. So if you have a paper bag and somebody breathes out, normally if somebody breathes out, all that CO2 goes out into the world. But if they breathe into the bag, it starts to accumulate in the bag, and then when they breathe in again, some of that CO2 goes back into their lungs, which means it goes back into their body and back into their blood, and it makes the pH go from a high pH back to a low pH because they don't breathe out as much CO2, because when they breathe out a CO2, it hangs out in the bag and then they're more likely to breathe it in versus if they weren't. Remember we talked about diffusion before. Molecules move a really certain speed unless they hit a barrier. And if they hit the paper bag, that's enough of a barrier that the oxygen and the CO2 get trapped, it bounces off the bag and it goes back into the lungs as that person breathes in. So as they breathe in from the bag, that traps some of the CO2, they breathe some of their own exhaled CO2, that causes the CO2 to start to form back into the H2CO3, pH goes back down to the pH of 7, and then they go back to the normal pH and their body is able to transport oxygen again. So that's an example of blood buffering being really, really important. Buffers are really important for biological systems. And one example that's really a good example of that is things like the buffering of blood, which helps us transport oxygen. So that's a good spot to stop for today. So thank you guys. That was the end of that 8th chapter. So the 8th chapter on acids and bases. Acids and bases are just special solutions that look at the amount of H plus or H3O. So H plus or H3O, those are when we're looking at an acid or a base system, that's the amount of material we're looking at. So these are just solutions of water with an extra component in them. That's the 8th chapter for this Top Hat program.